Hi, everyone. Welcome back. Welcome to the GATK Variant Calling Workshop, Day 2. I'm June here, and I'm going to continue the topic followed by what we discussed yesterday. So before we start, do I have any questions from yesterday? Like if you were able to run some of the codes and if you run into any trouble? Well, if not, let me start for now. Well, let's see. See a message. No. Okay, good. So yeah. So while I'm sharing my screen, I can't really see the chat box. So if you have any questions, just feel free to unmute yourself and ask a question. All right. So let's get started. So yesterday, we ended with discussing how we should prepare our BOM files in order to run GATK to call the variance. So by now, I assume most of you guys have prepared a few of the BOM files from the individuals that I provided in the practice data. So they should be ready to go. So now what we're going to start today is to actually talk about variant calling. So before that, I wanted to clarify on something. Someone asked me this question yesterday, and I think that's a great question. So we discussed mark duplicates, which is one of the first steps that we use to clean the BOM file. Like after we map our reads to the reference genome and we have our original BOM file, the first step we do is to mark replicates. So the question that I got was whether or not mark duplicates itself removes any duplicates. So the answer is this by itself, unless we specify it to remove the duplicates, it by itself does not remove the duplicates. What it actually does is it looks at your BOM file and it identifies the duplicates and it adds a sum flag to that read if it's a duplicate. So then the duplicates are marked and flagged, but they are not automatically removed. So in the BOM file that is generated after this step, like that ddub whatever .bom, so that file has all of your original reads except like the ones that are actually duplicates are marked up. So you can request a remove duplicates this option. So it actually depends on how your metrics output looks like from this step. You can look at your metrics and see the percentage of your reads that are duplicates. If it's not terrible, I don't recommend doing the remove duplicates because what it does is it actually would decrease the coverage. So if you look at your metrics file and see, well, like most of your reads are okay, there's a small percentage that are duplicates, what will happen is all of the downstream tools that are in GATK would like, when it's like using those reads to call variance, it will read the tag. And for those that are duplicates, it will just automatically ignore that by default. So by this, you don't like, unless your reads are terrible, otherwise you don't really need to worry about removing the duplicates, but it is being taken care of. So this is just one clarification from yesterday. I think that was a great question. Okay, let's start from now. So now that we have the cleaned and ready to go BOM file from each of the individual. So what we're going to do first is to get a GVCF file. So if you still remember from yesterday, the GVCF file is technically a VCF, but it includes not just the variance, but also includes the non-variance. So it's going to show you the genotype for every single location in your BOM file. So the GVCF will still be relatively large. It will still be smaller than BOM file for sure, but it will be like quite larger than the actual VCF file that it will get. So we're going to run something to get a GVCF per individual. So remember that right now at the first step, like for the GVCF, we're still gonna get one per sample instead of one per cohort. All right, so the function we're going to use in GATK to actually do this magic step is a program called haplotype caller. So this is the most essential part in our GATK variant calling, like the haplotype caller program. So what it does is it looks at your BOM file. It will identify regions where there are variants, and it's going to call them. So it will call both of the SNPs and indels at the same time. And also one thing that you may have noticed if you look at some workflow from between GATK3 and GATK4 is in GATK3, like some people, like there was one step of recommendation before you run the haplotype caller, which is you do some local realignment for your calls. And in GATK4, that step is incorporated in the haplotype caller already. So you don't necessarily need to run the realignments separately. So this step will take care of that, and it will generate you a file with reads that are reassembled. So if you look at a workflow of haplotype caller, it may look like more straightforward how this function works. So first of all, this is how your BOM file should look like. So you have all these reads stacked up for that are aligned to the reference genome. So what it's going to do is we know the allele in the reference genome. So for the haplotype caller, it identified these regions, what it calls active regions. So what that means is these regions that possibly contains the variants. So it identifies those, and depending on how the reads look like, it may work on some reassemblement. So for example, if you have some reads here that with a lot of like indels, the reads may look like not well covered for such a region. So haplotype caller would automatically reassemble these reads if necessary. So to figure out which regions are being reassembled, so we are actually going to specify in our function when we're calling haplotype caller using a -- BOM out option, it's going to generate us a new BOM file. So that new BOM file generated from the BOM out function is not, it doesn't contain all of your reads in your original BOM file. What it contains is only the parts that are reassembled through haplotype caller. So what you can do is after you finish this step, you load both of your original BOM and this BOM out. You load both of these two files to the IGV, that program, the Integrative Genomic Viewer, that program that we discussed yesterday. So you can load both of them to IGV, and you can literally scroll down the chromosome and see which parts are being reassembled. So I'll have a screenshot later on. So this is this part. So after haplotype caller decided like we clean out the BOM file, everything is ready to go, it is going to determine the likelihood of genotypes at every single site through an HMM, hidden Markov model algorithm. So I'm not going to dive into the details of how this HMM works, but all we need to worry about is after this hidden algorithm, haplotype caller is going to generate us the genotype for every single sites in our sequencing. So another question that I got from yesterday, now that we're already talking about haplotype caller, is why like this whole workshop only focus on germline variants, and why this whole process is not as good to identify somatic variants, which is the mutations that are not in the germ cells, but instead in any other body cells, for example, skin cancer. So I think it's also worth clarifying that here today. So in theory, if you think about identifying variants, in theory, the methods should be like if we develop a method that identifies variants, it should be good to call variants in any cell. But in reality, the principle of us calling different type of variants are different depending on what type of variants they are. So for the germline variants, or whatever we have talked about in this class so far is all we need to care about is whether or not the variants or the the sequencing calls are different from the reference. So if you still remember, every step when we're setting up, like cleaning the BOM, and also later on, at every step, we only specify the reference genome. So which means all we care about is if in the germ cells, if there's mutations that are different from the reference, we'll identify those as variants. For the somatic variants, or if you think about a cancer cell in particular, we not only need to care about the reference genome, but we also need to care about how those cancer cells are different from a normal body cells. So we call those controls. So we have the disease, we have the control. So to identify those variants are called variants when they're different from the reference genome and when they're different from your normal body cell like genome. So because of this difference, and also we know that for cancer, because of this, this is like mutation in the body cells, it may actually not just be like SNP or indel mutations. It could be something like even more dramatic. So for the germline variants, we know like what is a ploidy for our individuals. Like for human, we're diploids. But then if you think about a cancer cell, like since that's technically not a part of us, it may no longer be diploid. It could be, but it may not be. So we need to consider that in the variant calling too. So and also when we're calling variants in any other body cells, we're comparing things between one individuals, whereas when we're talking about the germline variants, we're calling variants jointly in like multiple individuals in a cohort or population. So because of this difference, like the principal difference in calling this different type of variants, haplotype caller is optimized and designed for just germline variants calling. If you use that, if we're set up for calling somatic variants, it may still identify some of the variants you're interested in, but it's not optimized for that. So we don't recommend interchangeably use this tool for different type of variants calling. So if you're interested in somatic variants calling, I recommend you reading some of the articles on the GATK website, which is really helpful, and the principle we learned from the germline variants calling, some of this will still be helpful for you if you're interested in somatic variants calling. So I'm not going to dive too much into that. I just want to give a brief clarification on why we only talk about germline short variants calling here. All right, so to initiate haplotype caller, we can simply call it directly from GATK like we did before. So to figure out what are the input for this, we can simply, like now I have logged in to a node on my cluster. I can simply copy paste this command like we did yesterday. Like we specified, like we load GATK module and Java module before, and we just need to call GATK and call this program directly to see what options it requires from us. Maybe I didn't load GATK. And let me load Java as well. Now, okay, it's probably some weird thing happening when I'm copy and pasting this from the text. Let's see if it likes it now. Yeah, it seems like it's working now. So when you're copy and paste commands, I recommend you doing that through like bbedit or any other text editing file, in case like on the slides or PDF, there's some things happening. So here we called GATK, and we can find haplotype caller there. Let me just type this thing because it looks like it didn't like some of the things we did. Let's see if it now works. All right, never mind. I have a screenshot in my slides. and I'll figure out that later. So if we just call GATK and call haplotype caller, technically, I don't know what is going in my commands now. I should probably just retype that like from the beginning. But anyway, it will give us a printout of what is the required argument. So we definitely need to specify what is our input. So the input is the BOM file that we cleaned already. So after we did mark duplicates, add group if necessary, and after we did the BQSR, we are gonna give that as the input for haplotype caller. The output is the GVCF. But if we don't specify we want GVCF, this by itself wouldn't give us necessarily the GVCF format. So if you just do input BOM file, give it a reference genome, and if you just do like output whatever .vcf or even .g.vcf, without anything else, it actually will give you a VCF. Like it wouldn't give you the non-variant site. So we need to specify that we want GVCF as our output because we are calling this per individual and we want to call variance among all of our cohorts. So we want to specify that. So there are two options we can use, and both of those will give us a GVCF type format. So we use -ERC, and if we specify GVCF, it will give us directly a GVCF. So the output, like the name of the file would be a VCF file. So the only way we can know like the output is a GVCF is when we're naming the output file, we write .g.vcf or whatever we want to call it, just so that you don't confuse that with the ultimate VCF that we are interested in. Alternatively, we can also use a different function or different option instead of like ERC GVCF, we can also use the BP resolution. So on the GATK website, it discusses further in detail what exactly is the two difference. Technically, they both give us a GVCF type file. They should both be useful, but just for the the best result, we'll prefer this option here. And also, if you're running our so for whole genome BOM file, these four options are all what you need to specify. For running our practice data specifically, since we're only using chromosome 20 from the 1,000 genome data, we want to specify that chromosome when we're running this. So if we don't do that, that's completely fine. It will still run without any issue. But it will make things slower because what haplotype caller does is literally walks through the reference genome from chromosome 1 all the way through chromosome X and Y. So if we don't specify the interval, like which chromosome we're doing, it will literally scan through the whole genome. So since we only have chromosome 20, it will start from chromosome 1 all the way through chromosome 20. And those, now since the data doesn't exist, it will just run taking the computational power without giving us anything useful. So for the practice for this class specifically, to make things faster, you can specify the interval we're interested in. So you can use -L and use 20 to specify that. So when we have everything set up, we should get a GVCF output. So this is a screenshot of the GVCF output. This step for just one chromosome, it doesn't take that long. So if you want to sit in front of your computer, watch how this is happening in live, what type of output log file like it will generate us, you can see that happening. For the practice data that we have for calling genotype on chromosome 20, it may take something between 5 to 10 minutes. So it's totally doable if you want to see that happening without submitting that as a job.
So she's never had.
I have a question.
Yeah.
So why do you give like the -O as well as the -ERC GVCF? I thought like one of them is enough to give the output, right?
Yeah, you mean here?
Yeah.
Yeah, so one of them is enough. Like okay, so the -ERC is just is an option. Like we need to give it an input after that. It's just like -I, we need to say it's a BOM file. So -ERC, we need to specify GVCF. So but one of, like, you can do -ERC GVCF, or you can do -ERC BP resolution. So
But
one of
we
these two is,
already have -O, right, in the command. Didn't we have -O, which is the output?
oh, the -O is just like, this is just to specify the output name. So by doing -O, you put G.VCF, it does not necessarily give you GVCF. You need to you need to specify you want GVCF. So this is just a file name.
I see. Okay.
Yeah. And we put a G here just so that we know this is a GVCF file.
Okay.
Yeah, so you can call it whatever you want. So this is just this is a string here. Okay. And this is, we're specifying we want the GVCF output. After we have the GVCF output, we can directly view that in our terminal. So this is so VCF is essentially a text-based file. It's tab delimited. So you can view that directly. It's not binary. So you can make that a like you can make a compressed GZ file, or you can do a VCF file directly, depending on how big of a file you want. In any case, you can just look at a few lines. So the start of the GVCF is almost identical to the VCF file, that it starts with a bunch of headers specifying the information for like how we got this file. And it will start with the column names for the just like the VCF format, like we discussed yesterday. So the only difference between this and a regular VCF is this column here. So at the alternate allele, because GVCF includes variance and also non-variance, so not every position will give you an alternate allele, because it just maybe just identical to the reference allele. It's totally okay, because we're asking for every single position in the GVCF. So you will see a lot of this block as non-variance. And that is okay. It just shows you where you're getting the non-variance, where your variants are. And to read the rest of the this file is the same way as how we read the other VCFs. So yeah, so like just an example here. So since here we have non-reference, it means like at this position, our individual has just a reference allele. So that's why the genotype is 0/0. So that's not surprising. This is what it's supposed to be, since they're all reference alleles. So we also remember that we ask the program to output us the reassembled BOM file. So you can name it whatever way you want, so you know that this part is a reassembled BOM file. So if you have questions about the GVCF, like, so say after the downstream analysis, you look at your VCF, you figure that there's maybe some part that looks fishy, or you want to make sure you're interested in a specific variance. For example, you like, there's a like whatever chromosome, you're thinking that at this gene, there should be a mutation that confer maybe positive selection in a population. You want to make sure that variant you identify is a real variant, it's not an artifact. So one thing you can make sure is you go back to the BOM file to see whether or not this variance is in a like a reassembled part, and if there's anything fishy about those reads at that particular region. So what you can do is you can get both the original BOM file you have, and also the reassemble this .out.bom, or whatever way you want to name this BOM out file. You load them to the IGV. So this is just one example of one part of our data. It shows that for this particular part of the genome, there is a part that the original BOM file may not look as well. So haplotype caller reassemble this part for this. So this is our original reads, and this is reassembled part. So you can zoom in and out and see like all of these reassembled parts. So this is just a one way to do sanity check later on. So otherwise, we should be happy by this moment with
So I have
our
a question.
Yeah.
Can you go to the previous slide? So you said this is a GVCF file, right?
Yes.
So GVCF file should have all the positions from 60,001, 60,002, and so on, right?
Mm-hmm.
So why have like there is 60,027 and
Yeah.
28
So the
is missing and 29 is there.
Right. So the reason why that happens is, okay, I don't remember the very details, but so if you count the number of sites in the GVCF, it doesn't match exactly the number of sizes. Like so, say the chromosome 20 is 57 megabase. If you count, it's not 57 million. So what happens is in the GVCF, it gives you this non-variance block. So I think it is it has something to do with the haplotype color that it identifies. Like if there's a whole chunk that is non-variance, it will record this as a non-variance block. I don't really know from the top of my head like how exactly that works, but I don't think you need to worry about like it's missing sites. It should have, like, as long as it's sequenced, it shouldn't be missing. But it's just like it gives you blocks of non-variance, and for the variance it will have them too. So with that being said, like if you're interested in the call at a particular site, I'm not sure if if that's not a variant, like you were interested in any position in your sequencing data, the best way you can go is to go back to the BOM file to see like how those reads align and what they are. The GVCF is more like an intermediate way that is like this file in particular may not be super helpful, but we want this to jointly call VCF eventually. So this is more like a temporary output that we need. We need this to call our variance. So I know in some projects you may be interested in the total number of sites, because we know the total number, like total length of the genome, but the actual number of sites that we sequenced that may be good is may not match exactly the total length of genome. So we call that callable sites. So if you're interested in estimating mutation rate, for example, you may be interested in the number of callable sites in your data. So if you want to estimate the number of callable sites, the best way you can do is to go back to your BOM file, because that will include every single position that is actually sequenced. So if you just simply count the lines in GVCF, it may not give you the right answer. So I think that's a long story of saying GVCF is a necessary intermediate output, but I don't recommend we look too much into that unless there's definitely something wrong with your VCF. You go back to the GVCF to see what happened.
Okay.
Yeah, but yeah, that's a great question. Okay, so now let's assume this step worked smoothly. We did that for several individuals. We have GVCF for every single one. Now what we want to do is simply join them together and identify the variance. So intuitively, if we think about how that process work, is simply like if we imagine we have GVCF for several individuals, we know those non-variance blocks. So we can think that this algorithm in GATK is just going to identify the non-variance among individuals. So if there are some sites that are non-variance for all of the individuals, they will not be included in VCF file. But if there's a given site that is non-variant in some of the individuals, but it isn't variant in the other individual, since we want to call variance among all of our cohort, those sites will be like such site will be included as variance in our ultimate VCF output. So it is not hard to imagine how this process really works. So specifically to actually run that in GATK, to jointly call variance across different samples, it requires 2 steps. The first step is simply to consolidate our GVCFs across the samples. So there are 2 ways you can do that in GATK. One is you literally join them together. So there is a function, I think it's called combine GVCFs. So that one, it will like literally look at all of your GVCFs among all the samples you have. It'll give you a humongous GVCF file. That process is really slow, and it really depends on the size of your genome and also the number of individuals you have. I really don't recommend using that. You can run that if you're curious about how that works, or if you absolutely need a GVCF that contains all of the individuals, but just a warning that that function really takes a long time, and it may not actually be helpful depending on what you really want. The recommended step to consolidate the GVCF is this function called genomics DB import. So this on the GATK website, they have a long explanation on what exactly this function is and what is the underlying computational algorithm or what it's actually doing. So long story short, what this is doing is instead of giving you an actual GVCF output that contains all of your samples, it looks at all of the GVCFs and give you a database. So the output of this process, instead of a file, it actually is a directory wherever you want to specify that. So if you look at what we specify under this function, is we call GATK, we call genomic DB import, we specify our reference genome, we give each our samples, like by the -v, we give it all of our GVCF files. So if you have 10 samples, you specify 10 times. If you have 3, you specify 3 times. And most importantly, we ask us to create us a database. So after this option, this genomic DB workspace path, what this is this is a string input, and this is the name of that database directory this is going to generate you. So for example, here I'm in my directory where I was calling the like I was calling the GVCF, and I was creating this joint database. So under this directory, after I did this process, it gave me this database. So this is a folder. You can cd into that and see what it contains. It contains some, well, it's mostly index files with, it's a database of like how your GVCFs look like, so that, I mean, this is not really readable for us, but this is very useful for the next step. So this is just a database that the genomic DB import creates. So if you go to the GATK website, if you're interested in what exactly this database is about, it talks about like, it uses like an Intel platform and what this database is related to. But anyway, for us, we just need to know that after we run this process, the output is a database that is in a separate directory. And again, in this part, we can specify our intervals. You can code multiple chromosomes altogether. You can specify multiple chromosomes here, or most of the time, what is helpful is we actually call VCFs per chromosome. So we can later on join all of these chromosomes together and have a whole genome file. But depending on what you need, a lot of times I find calling VCF on separate genomes very helpful, so that if there's particular genomic regions you're interested in for your work, you can look just those files without loading the entire whole genome VCF. So for this part, we can say that to create this database, we want this database only based on, say, chromosome 1. So we put the intervals as chromosome 1 here. So in our practice data, Here, instead of putting the CHR like 20, remember that yesterday we said that the way our contigs are labeled, they're just numerically, like from chromosome 1 through chromosome 20. So since in our like original index file, it doesn't include this CHR as a part of the string, when we specify it here in our real data, in our practice data, we can just say -- intervals 20, and that will do the job.
So when you say intervals, it will just look at that particular chromosome and leave the other chromosomes out of analysis?
So you can, yes. So if you say intervals chromosome 1, then this database includes would only based on chromosome 1, and it will ignore other chromosomes. So if you want, say, 22 chromosomes separately, so you just repeat this command for like chromosome 1, chromosome 2, and all the way through whatever chromosome that you're interested in. Alternatively, or not alternatively, additionally, you can also specify the interval as literally just a part of your genome. So say I'm only interested in, like for my research, I'm not interested in the entire chromosome 20. I'm only interested in, say, from the 10 megabase through the 50 megabase of the chromosome 20. Then here you can specify that as also specify as your interval. So instead of just saying 20 as my interval, you can say 20 comma, is that a colon? And say 10 megabase to like 40 megabase. So you can use that as your interval. So it really depends on what you're looking for. If you want separate chromosome VCF, you can just specify just the entire chromosome here. Does that answer your question?
Yeah, yes.
Okay, cool. So after we run that, we're ready to go for our last step, which is to genotype this joined VCF. So the previous step, genome DB import, even if it doesn't give us a specific file, this database contains all the information for the GVCF of all of our samples. So lastly, we want to actually get the VCF that we want. With that, we want this function called genotype GVCFs from GATK. So we call GATK, we call this function, again, we specify our reference genome, and the input, instead of a GVCF file, it has the database we just included. So for example, you name your database here, my database chromosome 1, or in my case, I call that like GBR, like chromosome 20, whatever you name that. You put, when you specify that in the genotype function, you put that string you created after this GenDB comma / like after this, you put the string you created like there. So this part, like -v, and this part is mandatory, and this is the custom name that you created for your directory. And then you specify the output. You want the output to be whatever name .vcf. And here you can also get a .gz VCF, like the compressed version. Either way should be fine. So this is all you need to obtain the VCF. And this step itself doesn't take that long. What takes long is this step. So if you're running a job, make sure you request enough time, because this step, since it is going through all of your GVCFs, this step will take quite a while. It depending on how big your file is and how many samples you have, it may take anything between a couple of hours to quite a few hours. So after all that, when that's finished running, you're ready to go for this part. So this part, if you're doing chromosome by chromosome, this step doesn't take that long to run. You can watch that happen. So again, here.
Could you explain what this step does actually?
This step?
No, the next one.
The next one.
This one.
So what it does is it now, like, so the previous step creates a database that contains information from all of the GVCFs. So this is simply given that database, we want the actual VCF file.
Okay.
So the input for this is reference genome at that database you just created. And the output is the actual VCF that we want. Like once we get that, we're good to go. Yeah. So this is a sample of how this output looks like. So this is just like the raw VCF that the way we read it is the same as how we explained yesterday. It contains all the header information. And here we can see that other than the first 9 columns that is shared in every VCF, we have the 3 individuals that we call genotypes for. So like on the terminal, this is how it looks like. If you want a like cleaner read, you can extract this file and open that in Excel, for example, or in R. And you can actually have this more like neatly showing, like each of them is a separate column. So we see that it should include all the variants, including the SNPs and also the indels. So say, for example, for the first position that we got, we have 3 individuals and 2 of them are 0 zeros. They are not variants in terms of like in contrast to the reference genome, but since one of the individuals is homozygous for the alternate allele, this entire site is included as a variant in this jointly called VCF file. So now, after this step, once you obtain this file, we technically finished GATK for variance calling. So the next step after this is with this raw VCF it's technically all we need, but not really. We want to do some more like sanity checks, quality filtering, and more refinement so that this VCF that we have is actually ready to go for our downstream analysis. So this is a raw output. This is like, you definitely should save this file. Usually the VCFs are not that big unless you have like a huge population. You want to save this file permanently. And whatever filtering you do is based on this file. So how about let's take a 10 minutes break. Let's come back here at, so right now it's 2:23. Let's come back at 2:35. So about 12 minutes. Let's take a break and then we're gonna come back here and we're gonna talk about how we do further filtering and calibration on the raw VCF we just obtained. All right, so I'm gonna, okay, welcome back. So let's continue. So let me share my screen again. All right, so I just wanted to show you what this VCF output from our from this previous step looks like. So in the VCF folder, here I have the original output that we generated from this step we just talked about, the genotype Gvcs file. So again, just a reminder, it reads the reference genome. The input is our database for the consolidated GVCFs across individuals. And the output is the VCF for the given genomic interval that we're interested in. So the interval could be an entire chromosome. It could be multiple chromosomes separated by a comma, or it could be a specific part of a specific chromosome. So anyway, so in my example, since we have chromosome 20, I specified chromosome 20 in the previous step. So here I called VCFs on the chromosome 20 on that a few individuals that I'm interested in. So I just wanted to show you how this file looks like. So at first, we see that we have a huge header part for the VCF that we have. It includes all of the information and also the acronyms for the VCFs you're going to see. And it also includes all of the commands that were previously used to generate this VCF. So that is really helpful so that if you give this VCF to anyone else, they will know which commands from GATK that you use to come to this VCF file. So it's trackable, and that is really useful when you're sharing VCF files across or between different colleagues. So we have a long config part information. Let's skip that. Here we go. This is the start of our raw VCF output. So we have our header. We see that we have the first 9 columns and we have 3 individuals. So, and in this part, this is this file only contains the variance instead of the GVCF that contains both variance and also the non-variance. So one thing that you'll notice here is this specific column, the filter part is all empty. So when it's empty, it's represented by a dot. The ID is empty because we didn't specify that. So that's no need to worry about as long as we know which reference genome we're using, like what is a coordinate, we can always find that if it's a SNP, we can find the SNP ID somewhere else. So that's nothing to worry about. But the filter, this is really important because we know that we just called VCF based on the bound. Hopefully everything here is good. But since calling VCF is essentially a like if you remember haplotype caller, it actually looks for the likelihood for genotypes. So since it is a statistical model, it allows it's almost like unavoidable that we will have false positives here. So this is a raw VCF we have. We hope everything here is all good, but in practice, things may not be that perfect. So some of the variants here may actually be false positives. Well, there could also be false negatives, but what we, I think what usually what concerns us the most is when we think there's a variant, we read from the VCF there's a variant, but it's actually not. So that will be the more concerning one. So we want to add information to the filters to look through the VCF files and use different ways to filter things and to figure out whether or not we want to keep a specific variant. So say if we create different ways to filter things, and if it's still a pass, then we're happy with that, we keep it. But if not, then most likely at least we want to flag it so that we know that in our downstream analysis, this is something to look out for. So this is like long story short, this is our raw VCF output. The next step is we want to further refine our VCF output to make sure that everything included here are trustworthy variants that are good for whatever downstream analysis we're interested in. So let's come back here. To refine the call set or to further filter the variants, there are essentially 2 ways where we can go to figure out whether or not there is false positives and how we annotate those and even further get rid of those. So before we go through any of the specific methods, what we usually want to do is, since our original VCF output includes both SNPs and indels, depending on your work, most of the times you want to have SNPs separated from the indels. Because like for example, if you study population genetics questions, a lot of times, if you just use the SNPs, that is enough to tell you whatever story you're interested in for whatever downstream analysis. And indels is a more interesting story because there are structural variants, there are more uncertainty related to like whether or not this is a true call or what we can exactly what we can do with this indel information. So we don't want to throw them out, but it's also better to separate the SNPs away from the indels, because for a lot of downstream analysis, it only requires SNPs. So we want to keep a SNP file and the indel file. So in GATK, this is something really easy to do. It is literally 1 line of code. So we can use a function called select variance. So we call GATK, we call select variance. The input is the VCF we just generated. So it doesn't matter whether or not a VCF is a uncompressed format or in the GZ compressed format. Either way, this function will take that in. So we specify that as our input, and we say we want snip or we want indel. And we specify an output name, and we run this code as is. It will give us a file, a VCF file that is just with snips, or VCF file that is just with indels. So again, for our output name, we can specify that as a VCF output, or we can specify that as a compressed version of VCF. So either way, that will work. So just give you an example. I run those two codes respectively, and I generated two VCFs based on, like, it's not new VCF, it's just subsets of our original VCF. So I have a snip VCF and I have an indel VCF. Let's just take a quick look at how they look like. We can just look at the tail to skip the headers. So here we see that nothing changed compared to our original VCF because all we wanted to do is to subset them. But if you look at the alternate alleles, you should have just the SNPs. So you shouldn't have multiple letters here because we're excluding the indels from this file. So this file is just includes the SNPs. And if you look at the indel file, again, let's just look at the tail. We see that every position included in this file is either an insertion or a deletion. So in this case, this is a reference genome and this is alternate. So this is a deletion. And in this case, we have an insertion. So usually you should have most of your, like if you look at your original VCF, most of your positions shouldn't be structural variant. So the indel file should be fairly smaller than the SNP file. So this is just how we separate them. Okay, now we have them separated. What do we do with those? There are essentially two ways where we can filter or like reevaluate our variants. So one way is very similar to the BQSR that we talked about yesterday. So if you still remember what BQSR is that is a process where we want to refine the BOM file calls. So what we did is we want to make sure our BOM file is okay. So to do that, we train a machine learning model using the true sites. So if you remember that, we specify some non-sites where we give them the 1,000 genome indel files and also the 1,000 genome SNP sites. So those are validated from previous studies. Those are high quality sites that we should trust. So the machine learning model are trained based on those non-sites. So it learned which sites are good, what type of sites are bad. So it associates the sites with the quality scores, the base quality score in BQSR's case. And then once we have a trained machine learning model, then the input file is the BOM file that we had, and it gives us a recalibrated BOM file given that machine learning model. So for evaluating and calibrating the variance, we can use a very similar method. So it only varies by one letter. So instead of base quality, we call this variant quality filtering. So it's we call it BQSR. BQSR can happen through the variant recalibrator function. So this is essentially identical with that base calibrator function, except this is instead of the input being a BOM file, the input here is going to be VCF files. So what we do is we again specify a set of non-sites, and the more database we give it, the more accurate this machine learning model will be. So we specify a bunch of non-sites. We'll need to download those database beforehand. We feed those into the machine learning model, and it trains the model, and it takes our input VCF file, the raw VCF that we just got, and it's going to assign a log-odds ratio, it's a score, to each of the variant. So after we do that, it will generate us a recalibration table that will tell us like which size may be off. So similar to the story of the BQSR. So once we have that model, then we can apply the VQSR to our VCF to get a new VCF file. Okay, so that's a long way of saying how it looks like. This is specifically how your input should look like. This looks really crazy, and I'm sorry about that. There's no way around it. So let me try to break this down to what exactly it is. Again, the best explanation for what exactly each of these function does and why the input are set as they are, the best way to find that out is to go back to the GATK pages. There are really detailed explanations out there that will be very helpful for you to understand what exactly these inputs are. So long story short, we call GATK, we call variant recalibrator. We specify this is our input that we just got, that VCF file. And we have a whole part where we're trying to ask, like, we don't really need to change it. Like you can copy paste that from the GATK website or from here. This is pretty much saying what are the threshold this machine learning model should be. Like how we should set this up, what type of model you should use. It pretty much uses a Gaussian mixture model to set up this as the algorithm for the machine learning. So this whole part, we don't really need to change that. Well, there is some difference depending on if you're training this for SNPs versus if you're training this for indels. So you can refer back to the GATK website to see what this setting is. So this whole part, you don't really need to worry about that. What you do need to worry about is first, your input, and second, what your output recalibration table, what you want to name that. And most importantly, we need to specify the training data for the machine learning, which is the list of known sites. So this part of the code will be very long, so I shortened this part. So for this part, for the resources part, this is what our training data set would be. So you're familiar with this one. We used this one yesterday. So since we're talking about SNPs, I use like just a SNP size here. So this is our 1,000 genomes SNP, the high confidence SNP data set that we previously downloaded from the Broad Institute. So we can use that as one of the training set under these resources. In addition to that, like that, that is good, but that's probably not enough. So there's several other databases that we can use as training set. So the story here is the more training data we give it, the more accurate this machine learning model will be. So if we downloaded multiple SNP database from the Broad Institute, we specify them here, and also conveniently by GATK, it give us this prior settings that what we should use for each of this respective database. So so for humans, we can use exactly what they gave us. We just need to download this database from the online resource, and we specify this here. We plug this part in to our code, and we run this thing as is. So the code itself will be fairly long, but if you think about what it is as simple as we call this function, we specify our input, we give it all the training set, and it will ask us to give us a recalibration table output. Once we have that table output, we apply this output to our input file. So here it's a similar story as the BQSR that we mentioned yesterday. We say that now this is our input. We want a new output VCF that is calibrated, and we can specify the filter, the sensitivity level. You don't have to change it here, or you can play around with this to see what happens. And most importantly, we want to give it the recalibration table that we just specified. And we say we're doing this for SNPs, not indels. and it will give us an output. So with this is going to take a little bit because we need to train the machine learning model. But in theory, it should give us a fairly accurate or trustworthy VCF that we can use for downstream analysis. It sounds really cool and it sounds really straightforward, but practically speaking, not every organism would be like applicable for this VQSR process. The reason why is because to run VQSR in GATK, it requires us to have all of this high quality, high confidence, non-variance sites. For human, we have that already. We have all different sort of database from like 1,000 genome, from HapMap, from like the DB, SNP database. Like we have that already. For maybe a couple of other model organisms that is frequently studied by scientists, we may have that too. But for most of the non-model organisms, like if you're studying, say, horse, we may not have this known size for that species you're interested in. So how about, like, you're not studying human? How do you filter this? You can't really train a machine learning model because there's not enough training data. In that case, we use a different way of filtering the variance. We call this hard filtering. The reason why we call this hard filter is because we simply set a flat threshold for every filter options that we can possibly think of. So there are, like, later on, I'm gonna talk about like what type of what type of filter we can use. But the reason why it's called a hard filter is we just simply said, like, say we want a quality score to be less than 30. So that 30 is a flat threshold. We can change that to 40, to 10, to 100, but either way, that is just one specific number. So it's not as flexible and well-tuned as a machine learning model, but when that is not applicable for your own research, hard filter is one way to go. So we set this, you can think as arbitrary, but it has a justification for this threshold. We set different thresholds for different criteria, and we apply those to our VCF file to give annotations to each of the sites. So, or more specifically speaking, we want to now we want to label the filter column in the VCF file. So right now in the raw file, that filter column is all empty. We want to actually fill that in, whether or not we want to say a specific site is a good site, or a specific site is fishy, depending on the criteria that we use. So to do hard filtering, we're going to use a different function in GATK called variant filtration. So GATK actually give us some best practice recommendation. So given the previous studies looking at how variants typically look like in terms of different filtering criteria, GATK overall give you a best practice recommendation of what you can specify here for SNPs or indels. So we're gonna go through each one of them specifically, but just wanted to give a head up here that there is a starter recommendation. Of course, every single one here is subject to change depending on how your data actually looks like. But when you have no idea, this is like the first step to go. Like, you can use the GATK best practice recommendation filtering criteria, and then you look at your output to see if that's what you want, and if that still looks good. And if not, then we can play around with the specific settings here. So GATK has that for indels and for SNPs. If we apply that, we'll generate a new VCF that with the filter part filled in. So before we go to the details of what each of them are, let's just look at how the code is set up. So let's look at SNPs, for example. So we call GATK, we call variant filtration, we specify our input. So in our case, since we separated SNPs from index already, we can just load the SNP file for the SNP specific filtering. So after we figure out what is our VCF input is, we want to figure out what are the filters. So what this is in our VCF, let's see if I can see that here. It's actually, they're all included in the info column. So if you expand one cell in the info column, you will see that that info column would include some of the statistics, including the quality score, including some other stuff that we later will talk about. So what it is this function will look at the info column in our VCF that we give it in, and we can ask that to filter things based on, so say there's a statistics called quality depth. We can say that, and in the info column, that quality depth is under the acronym for QD. We say we want to filter things based on the quality score less than 2. So it then looks at all of the sites, figure out which sites have quality score, quality depth less than 2. And if there are such things like that, we assign a name in the filter column. We call that QD2. So this name, we name that, but this is like a Boolean, like it looks at true or false given all of your sites. So all of your sites have this statistics. So for all of the sites that didn't pass this filter, it will end up with this name in the filter column. So then we can also say we want another filter, which is the quality score of the site. So then this function scans through your VCF, look at all of the sites that would quality score less than 30, and we're going to assign them a filter name. We call this, like we can name qual 30 or whatever we want to call it. So for a given site, after all of this, after we figure out how many filtering criteria we want to put here, all of the sites in the VCF would ended up with something in the filter column. So if there's a site that passed all of the filters, like it didn't end up with any of these flags, then those sites would ended up with a pass annotation under the filter. So whatever after this hard filtering, anything that was passed, in theory, you can keep them safely for your downstream analysis. Then for those that didn't pass, it will ended up with the reason why it didn't pass. So you it may pass all the other filters except the quality score. So then you'll see, okay, these two sites didn't pass the filtering because of the quality score was too low. And then you could also end up with some sites that didn't pass because of several reasons. So for example, this SNP didn't pass because it has too low quality score and it has too low of this quality depth. So you can end up with several notations under the filter. So in theory, after this practice, so this variant filtration function runs really fast because VCFs in general are just small. So you're pretty much running a bunch of like Booleans to them to figure out which one you want to keep. So let's see. This is a screenshot of how my VCF would look like. So if you just download this VCF, you can safely just say, I want to keep all the sites that would the pass in the filter column, and everything else, I don't keep that. So you can do that. So this is hard filtering. And once you figure out that, everything that passed, you can use them for your downstream analysis. So now the last part we want to talk about is what exactly are these filtering criteria that we used. Because just from the acronym, they don't look like anything. And why we ended up with this specific numbers that GATK recommended us to use. So the first one is what we call quality bed depth. So it is very similar to the quality score, like this qual statistics that we'll find in the info column. The difference is this is you can think it as a standardized quality score. that is, you take the quality score and then divide it by the read depth in the non-reference alleles or in the alternate alleles. So it is essentially a quality score, but it is standardized by the read depth. All of these plots I'm going to show here, I didn't make those. You can find them on the GATK website under hard filtering page. So what it is showing is, it is showing us why for SNPs, for example, we ended up with like the best practice is quality score less than 2 is not good. The reason why is because if we aggregate many studies together, like we have a lot of VCF files, we just wanted to see overall how the quality by depth this statistics looks like. We see that most of the variants have this QD between this range. So there are 2 peaks, and the 2 peaks of QD corresponds to the 2 type of allele you have. One is the heterozygous, because since we're only talking about variants here, it's either we have heterozygous, the 0 ones, or we have the 1 ones, like the alternate allele homozygous, or the non-reference alleles. So we have most of them distributed like in this range, and we have a small set of a sample that was very low QD score. So given this distribution, and also given the VQSR result, so this is in human data that we can also train a machine learning to see what our original data set looks like. And since in that machine learning, it will reject some of the sites from passing the VQSR, we can see what's the distribution of the ones that failed the machine learning training. We see that these two actually agree with each other fairly well, that in the VQSR, most of the things that didn't pass have a quality by depth around 2 or less. And also, if we just look at the original distribution, we see that there's a small set of data that would quality score less than 2. So given these 2 distributions, we arbitrarily said, well, we say maybe quality by depth, the QD being less than 2 is for sure something not good. So this is like one filter that we figure out like we can use, and we figure out where this 2 comes from. So similarly, we can use other summary statistics to figure out how to further filter things. So there's another one called Fisher strand. So if you remember the Phred scale probability, so that is in the VCF file. Like if you look at like each genotype, so at first you have genotype, you have read depth, you have maybe a allele balance, and eventually we have a probability, this Phred scale probability, like a log ratio, log scale probability about like what is the probability of this site being reference homozygous or heterozygous or alternate homozygous. So the one with the lowest score, which means they have the highest probability, is the one that we assign the genotype for. So for that Phred scale probability, we can use a Fisher's exact test to see like how good that genotype call is actually is. So ideally, we want the Fisher exact test to be like to tell us like it is like a good distribution between the alternate and reference allele. So ideally, we want this statistics to be 0. So if we have too high of this value, it means that specific site may be a false positive. So if you look at the distribution of these statistics in our raw data, and we also look at the ones that didn't pass the machine learning training, we figure that like, or GATK figured that maybe putting this at around 60 is a good like threshold for SNPs. And for indels, because indels are uncertain, like structural variants are hard to map, they're hard to call in the first place, we can put an even higher threshold for the indels. But in any case, we figure that like FS being higher than 60 is a good threshold to filter some of the SNPs. And another similar thing we can use to figure out this allele bias is this statistics called strand odds ratio, SOR. So this is just another way of estimating this strand bias similar to the FS. The only difference is this one, like this statistics may perform even better at like when you're outside of the exons. So similar story, we look at the distribution of the statistics and we look at the ones that didn't pass the machine learning. They decided that at value of 3 SOR is a good way to filter things out. At least this is not too aggressive, but also not too conservative. So things that are higher than 3 may be problematic, and you want to flag that in your VCF. Another thing is related to mapping quality. So this is related to like in your original reads, like how mappable this region is and what is that mapping quality score. So you can use the mapping quality score directly, and you can use the like a square root of the average of the, okay, this is a long way of saying, they call it square root of the average of the squares of the mapping quality score, or they call that root mean square. So you have this RMS mapping quality already included in the info column. So again, here, what it does is it looks for the mapping quality overall and also the standard deviation, and things less than 40 may be actually too bad. They may not be in a like well mappable region. In that case, at least you want to flag your variance. So the rest of this filtering criteria is a similar story. You can read it by yourself. They overall just look at they look at different ways, like different ways that a variant calling could go wrong. So over all of these statistics, we may end up with a set of variants that we think, since it passed all of them, they may be good. So this is hard filtering. So for our practice data, let's see what time we have. For our practice data, we can, like, if you're interested in the machine learning based method, we can, like, I downloaded those sites already under the non-sites folder. So you can copy those to your directory and use the VQSR, or alternatively, you can use the hard filtering. This step may be maybe faster than before. Either way should work. So what I want you guys to do after this workshop is we get a VCF that is called correctly on a set of individuals, and also is filtered respectively depending on which filtering method you used. It shouldn't, like, depending on how many individuals you get, since we only have chromosome 20, even if you include all 8 individuals, it shouldn't take that long to run. By the end of Sunday this week, I want you to email me the final VCF that you got. You can email me the entire VCF, or you can give me a SNP and indel separately. Either way, I don't really mind, because those VCFs should be fairly small. I think they should be like reasonable size to email. So just email me the VCF that you got, and also with either if you use VQSR, you have the problematic sites filtered out already, or you can literally label them in through the hard filtering. Like, I can see them from the filter column. Either way should work. So email me the VCF that you got, and also email me the set of commands that you use to generate the VCF. So it is going to be similar to this bash script that I shared with you yesterday. Just give me like a set of like commands that I used from the beginning, like how you process the BOM file, how you call the VCF, just so that I know it is traceable. And you can annotate those commands respectively. So just send me these 2 files. And if you're getting graded, this will be depending on how your VCF looks like. If everything looks good, you're going to get this, the grades for the assignment part. Today, shortly after we finish here, I'm also going to send a Google form link with a quiz. There's going to be a few multiple choice questions. It should be fairly fast to finish those. So if you're getting grade for this class, please finish this Google form quiz before the end of Sunday. So both the quiz and the assignments will be due by the end of this week. So with those and also with your attendance, you're going to get the grade for this workshop. And if you're not getting grade, you're just here, sitting here to just learn in general how GATK is. Those are not mandatory. But I think at least the assignment may be helpful for you to get some hands-on experience with seeing how the GATK workflow works. And those hopefully will be helpful for you in your own research. So with this, I think by now I covered all of the material that I want to cover for GATK. Do I have any questions?
So we got all these VCF files and stuff. What exactly, how do you use these files? Like what purpose do they,
What do they do?
Yeah.
That really depends on what type of research you're doing. So for me, for example, so say I have the VCF from 2 populations. There are a lot of things you can do with that. You can compute allele frequency distribution. You can look at like compare the 2 populations. You can do like ancestry analysis. You can do I mean, it really depends on the specific research question you want. But I would say most of the, like if you're interested in populations, most of the questions, like or the downstream software you're going to use, the input is going to be VCF, because ultimately we don't really care about the non-variance between individuals or populations. So like, I don't know if I can give like a specific example right here, but like what type of research question do you have?
No, I'm not working on variant calling or anything. I'm just asking generally. Like,
yeah, so I mean, let me think of an example here.
because not all SNPs give rise to disease. I mean, only some of them can contribute to disease. So how
yeah,
do
so
you go
for
about all
disease,
this?
for example, so say you have a so say you have 2 populations. You have one population, or you have 2 groups of people, one group with the disease, one without the disease. So you wanted to know like what contributes to like this maybe the higher disease risk. So what you can do is you have the VCF from both populations, and you can do selection scan, which is some programs that identifies like positive selection signals. So if in a population where they're more robust to the disease, maybe there are genes that like mutations in the genes that would confer a higher or lower susceptibility risk. So you can, usually the programs that do that takes the input as VCF instead of BOM file. So then you can identify variants that are either like change the fitness in individuals, or you can even run like a GWAS analysis to identify variants that are associated with the phenotype you're interested in. So yeah, it really depends on the question, but I would say most of the downstream programs, the input is VCF. So really this is where you're starting at. Like you have the VCF, and the reason why we do all this filtering is because we want those variants to be real variants. And then once we have that, like that can be used for literally any downstream analysis you want.
Okay.
Yeah. So yeah, I think once you have specific research question, I mean, you can like you can see like what programs you're required to use, and I will say like that's when your VCF comes useful. Like what yeah, if that answers the question.
Yeah.
Is there any other questions that I have? So for GATK specifically, like before you turn in the assignments or quiz, if you have more questions related to like if you run into errors, you don't know how to debug that, feel free to email me. You guys all have my email address, and like we can figure out those problems together. So after workshop, if you like, so say you take this workshop, you kind of get an idea how GATK is like, and later on if you have questions, I will say that the GATK website from the Broad Institute is really, really helpful. So for myself, most of my questions I got answer for from the websites. They have a lot of useful materials, like articles and also discussions on the common errors you'll run into. So I guess overall, this is a really mighty tool with a lot of and we're only covering the basics of what GATK does. There are a lot of other things it can do that I can't cover in this workshop. So hopefully this class gives you a overview and introduction to this useful program, and hopefully it is useful for your research later on. So yeah, so if I don't have any additional questions for now, I think I can just end the recording here. Thank you guys again for attending this workshop, and like I'm looking forward to seeing your assignment.